Me?
Morning, sir. Yes, we can.
Yes, sir.
Morning,
Good
sir.
morning.
Good morning. I think this is where we left off, and maybe we'll get started right here. Okay. Okay, I was telling you about Franklin D. Roosevelt, who had polio before he, as an adult, and then he went on to become the president of United States of America. And what I did not tell you in the last class is that he was instrumental in creating a foundation, a foundation called, I think it was called something like National Foundation for Infantile Polio. And then it was changed to another organization called March of Dimes. And what he campaigned that every American donate 10 cents and that can fund polio research. And it was a fairly successful campaign. And that led to actually the funding for eradication of poliomyelitis, pretty much done from most parts of the world today. Now, what is the principle? What are some two basic types of vaccine? We'll look at vaccinology in a separate class with details of more recent advances in vaccinology, including the ones that pertain to SARS-CoV-2, which includes at least two new technologies, and one more is in the pipeline for that. But that we will look at in a separate class. In today's class, what we look at is we look at what are the fundamentals of poliovirus vaccination. This is pretty much, for the most part, most vaccines that we know of fall into one of the two categories that we're going to discuss today. Though there are only very few exceptions that don't fall into these categories. Now, how do you create a poliovirus vaccine? So this was a major challenge that was put forth almost several decades, 7, 8 decades ago to scientists. And what they did was they took poliovirus. All of you know that there are 3 serotypes from the last class, and they inactivated it by adding some chemicals such as formalin. So what these chemicals do is formalin, like all of you know, is used to preserve anything that you want. It'll preserve the morphology, it'll preserve the architecture, but it does some cross-linking of, for example, DNA cross-linking, protein cross-linking, in a way that they don't really function the ways they're supposed to, and that is what inactivates the virus because of adding these chemicals. There are other chemicals other than formalin that can be added to inactivate a virus, and the inactivation is primarily by cross-linking different entities inside the virus, including nucleic acids and proteins. Now, what happens when you inject? See, this is called a intramuscular or injectable poliovirus vaccine, or IPV. This was the first vaccine that came out for poliovirus, and it was a very important step those days because people who were vaccinated did not get major illness. They were protected against the major illness. Anybody wants to remind me what is the major illness in poliovirus?
Paralysis.
Paralysis, and the two forms, if you can remind the class about it.
Spinal and bulbar.
Bulbar form, sir.
Spinal and bulbar forms, right? Those are the two forms, right? Now, this vaccine could prevent people from getting it. So what happens? Does anybody want to tell the class what happens when somebody takes an injection somewhere in the deltoid, somewhere in your arm? If you take an injection of a vaccine, what happens after that? Let's assume one of you took this vaccine, injectable poliovirus vaccine. What happens after that in our bodies? Anybody wants to try and explain that in like very simple steps, 3, 4 lines? Nobody wants to make an attempt? Okay,
So,
go ahead.
so maybe maybe the macrophage in the muscular tissues will like engulf these kind of viruses.
Okay,
I'm not
okay.
aware about it. Yes, sir.
After that, okay, so maybe I'll just run through it. It's partially correct what you said. So let's say this is the virus. So you get it injected in your shoulder or your deltoid muscle, and then what happens is there are cells which are referred to as APCs. What does APC stand for?
Antigen presenting cells.
These are called antigen presenting
Antigen.
cells. They are usually macrophages could do it. There are tissue resident macrophages, they could do this. There are other cells called dendritic cells which can also do it. So they are present throughout the body, and what they do is they are able to engulf these organisms, these viruses, or they may even bind to a receptor. That is also possible. And once that happens, what they do is they process the virus. For example, here the polio virus, it's going to be processed by these antigen presenting cells. By processing, I mean they break it down into peptides and hold out the peptides on their surface. And these antigen presenting cells, when they hold out these virus peptides on their surface, they call for T cell help, and they attract T cells and B cells. Okay. And these T cells and B cells, when they come and they recognize these peptides, which are a part of the virus, they get activated. And with T cells and B cells, there's a lot of interplay between them. I'm not going to talk about it. But at the end of it, maybe at the end of 1 week, what you have is you'll have antibodies against various peptides of this poliovirus, because it will have hundreds of epitopes. You'll have antibodies against all the hundreds of epitopes. Depending on how immunogenic each epitope is, you'll get antibody response against all the epitopes of poliovirus. You'll have that in your body. So that is usually going to be present in your blood. This kind of immune response, you'll have immunoglobulins or antibodies against multiple epitopes of poliovirus in your blood. Within about a week to 10 days, you'll have antibodies. Now, you just don't have antibodies, you have a lot more than that. Your T cells and B cells in your body, which are the most important immune cells for adaptive or specific immune response, they get primed, and there will be a reserve of memory T cells and memory B cells in your body hiding somewhere. They are very long-lived cells. They can live for several years, and they can remember these epitopes of poliovirus, and they live in your body for several years. So what do you get at the end of this vaccination? Any vaccination, poliovirus is just an example. You get at least 3 tangible things. One is you get antibody response. Second is you get memory T cells, various types of T cells. We won't get into that. And you also get memory B cells, which are the cells that produce the antibody. So these memory cells can remember. They almost have something like, if you can relate to it, something like a photographic memory of the virus peptides or the virus epitopes, antigenic epitopes of the virus. And next time you're challenged with a real virus, they can mount an immune response bypassing several steps, and they can do it in just 1 or 2 days, such that you can actually control the virus before it does the damage or it causes the disease. They're able to respond very quickly to the actual virus challenge, bypassing many normal steps of the immune response, because they've gone through it already because of the vaccination. They have a photographic memory of the virus or the virus epitopes, and what they do is they can respond in quick time and fight the virus. So instead of taking 10 days to respond, they can respond in a day or 2 days, because they bypass several steps, these memory B cells and memory T cells. And that's what happens when you take a vaccine. Now, if somebody
So I had
takes
a question.
Yeah, go ahead, go right ahead.
So what determines the approximate time scale of this like memory? Because from what I've heard, like some diseases have an almost a lifetime immunity, and some have like say 2 to 3 months.
Yeah.
So what
Yeah.
determines that sort of time period?
So actually, usually you don't have anything as short as 2 or 3 months, but it's, I think you're talking mostly about coronavirus. And because the antigens are so different in alpha, beta, gamma, delta, the chance of reinfection is a true possibility, even if your memory cells are actually functional, because they mount an immune response against something slightly different. But overall, what you said is correct. The longevity of memory cells varies from disease to disease. And what determines that is really not known. But overall, what is known is it depends a lot on the immunogenicity of the epitope or the neutralizing epitope. What do I mean by that? So there are thousands of epitopes. Each protein can have hundreds of epitopes. Like an epitope is usually like a small peptide, a few amino acids, maybe 10, 20, 30, 40, 50, in that range of amino acids. That will be an epitope against which the body mounts a specific immune response. Each virus has hundreds or up to thousands of epitopes. Now, only a couple of these epitopes, maybe 2 to 5 epitopes of the virus, they can elicit what we call as neutralizing antibodies. Okay, so among thousands, only maybe 1, 2, 3, 4, a small number of epitopes elicit neutralizing antibodies. So a lot of how the body is able to fight back when you're actually challenged with the virus beyond 5 years, 10 years, 15 years, 20 years, a lot of this depends on how immunogenic these neutralizing epitopes are. And what I mean by that is how good an immune response they're able to elicit, how strong an immune response they're able to elicit. Okay, so that is the most important determinant. Typically, what happens is lipids are very weakly immunogenic. DNA can be more immunogenic, but much less than proteins. So proteins are the most immunogenic. Among proteins, depending on the epitope and the secondary structures, the ability of the epitope to elicit an immune response varies big time. It can go to like 3 or 4 orders of magnitude. What do I mean by that? Let's say I have an epitope A from virus, let's say just poliovirus. Let's say it elicits on a scale, on an arbitrary unit scale, it elicits antibody response or immunogenic response of, let's say, 100. There could be another epitope of another virus that does 10,000. There could be a third virus where neutralizing epitope does 10 only on the scale. So that appears to be the most important determinant of how well the body can mount an immune response and respond to it. So it's largely dependent on the immunogenicity of the epitope and how good an immune response it elicited when you got the vaccine. And that has to be neutralizing also. So it comes back to the ability of neutralizing epitopes or the epitopes that induce neutralizing antibody, how good an immune response they mount, and that can be over several orders of magnitude, and that determines how long your protection is and things like that. But there could be other determinants which we don't know, because these memory B cells and memory T cells are very complicated. We have not understood a lot of things about memory B and T cells. But right now, as of what we know, it's primarily the immunogenicity of these epitopes. Does that make sense? Okay.
So one
Now,
more question.
go right ahead. Yeah.
So the response, that is the neutralizing antibodies, would be similar in all the people, as in different people we inject the vaccines. So would the neutralizing antibodies be same for them?
In all the people who are vaccinated. Is that your question?
Yes, sir.
Okay. Okay, let me answer it like this. Okay, let's say we give 100 people injectable poliovirus vaccine or SARS-CoV-2 covaxin, for example, which is injected, and it's an inactivated virus vaccine, just like this. Let's say we give 100 people one of these vaccines. So, like in general, the extent of neutralizing antibody produced will depend on whether you gave IPV. It may overall, on average, the immune response may be different for IPV versus the immune response for SARS-CoV-2 in terms of neutralizing antibody may be different. Overall, if you take an average, it will be different. One may be stronger, one may be weaker. Nobody knows which one is stronger. You have to do a lot of experiments, but they're not going to be necessarily equal. They can be different overall. Now, if you look at individual specific differences, let's say it's possible that out of the 100 people, 90 people responded very well, 10 people did not respond well, 5 people did not respond at all. So there is going to be a spread of response among individuals, and this is dependent on the genetic makeup. What does this depend on? This depends on a few things. Just because you asked, what does this depend on? This depends on the MHC, or the major histocompatibility complex, which is a cell surface molecule that's important for the T cells and B cells to recognize epitopes. And each of us have divergent class of MHCs. There are like more than 1,000 different variants of MHCs. And depending on what we have, our immune response may vary. That's one thing. Second thing is there's something called VDJ recombination, which induces diversity in the antibodies. Or for example, there are like one particular T cell, one particular B cell can only produce antibodies of a certain type because there is some splicing and there's some fancy stuff that happens that makes an antibody of a particular shape that will neutralize your SARS-CoV-2 spike protein, and the ability to have that kind of T cell varies in individuals. So if I don't have, for whatever reason, a T cell that can actually produce antibody against that, I will not respond well to this vaccine, or I may not respond at all. So individuals, if you take a population, individuals respond very differently to antibodies. Typically, to most vaccines, about 90-95% of the population responds very well. If the vaccine is good, 90-95% of the population responds very well to most vaccines. There's a small percentage of about 5% of people, even if the vaccine is very good, they don't respond because of their genetic makeup, which includes MHC classes or the variation in that, and the diversity differences in the T cell and B cell repertoire, which is the VDJ recombination or some sort of recombination. I won't get into details of that. Differences in these two aspects make different individuals respond differently, but about 90 to 95% of the population will respond well to most vaccines if the vaccine is good. Is that
Right, sir.
okay? Yeah, go ahead.
So since we were talking about the longevity of memory cells, so we all get vaccinated for various diseases, chickenpox being one of them. So what goes wrong? Because like I've heard many people contracting the disease even despite the vaccination.
Yeah, chickenpox, it's a good vaccine. If you have 2 doses of it, you will usually not get it again. If you had the full course of vaccination, chickenpox vaccine, like 95% people will respond well. They'll be resistant to any form of disease after the vaccination. Only about 5% of people may have the chance to get infected after the vaccine. So that is one thing. The second thing is if these vaccines are not stored or administered well, that can be an issue. But typically, as best as you know, chickenpox vaccination efficacy is, I think, upwards of 85 to 90%. It's a good vaccine. Did you get chickenpox after getting vaccinated?
Yes, sir. That's why I asked the question.
How many doses did you have?
So I guess I had completed my doses.
So did you have a specific diagnosis of chickenpox by a test?
Yes, sir. It wasn't very severe, but
No, but did you have a test to show that it is chickenpox? No, right?
No, the doctor.
It's a clinical diagnosis.
Just the clinical diagnosis.
Yeah, so that could be an issue because there are other diseases where you can have vesicular lesions which are mild. There are other infections which could be. So unless you have a clinical a laboratory diagnosis saying you had chickenpox, you can't be too sure. But typically chickenpox is a home diagnosis, or your mother diagnoses it, or or your basic clinician diagnoses it just by looking at you without doing any tests, right? So a lot of times actually that could be also wrong. It could be
Right,
caused
sir.
by other viruses. So that could be a possible reason. I'm not saying it never fails. I'm saying it doesn't fail in a large number of people. That's all. Okay.
Yes, sir.
Okay. Okay. Now, these individuals will have antibodies in their serum. They will have something called. Anybody knows what class of antibodies these people will have? They'll have IgG class antibodies. For those of you who are not familiar with, how many of you do not know the 5 different types of antibodies? If you don't know, just say yes or no. Just say no if you don't know.
Sir, we have not done like a course on immunology, so there is basic, very basic things we know about these things. So if you, it will be better if you can elaborate whenever
We studied
these
them in
terms
very
come.
basic during SBL100.
That's right. I taught you. I teach this in in great detail. Anyway, so maybe this. Okay, I'll just use an acronym. Maybe I'll just give me a second. I'm just going to erase everything. So for those of you who are not familiar, there are 5 different classes of antibodies. I'll use an acronym called GAMED. GAMED. These are the 5 different classes of antibodies. So you don't have to necessarily remember all these, but I'll tell you some basics so that you know what happens. Anytime when you're exposed to a particular pathogen for the first time in your life, you get a vaccine for the first time, you're exposed to a particular antigen, you get an IgM response. Within 2 weeks or 3 weeks, the body switches that to IgG. You'll only have IgG in your blood against that pathogen. For years together, you'll continue to have IgG in your blood, right? How long you have it varies from pathogen to pathogen, but you'll have IgG antibodies in your blood for very long periods of time. Without going into the other types of immunoglobulins, which is, I spoke about IgM, which is anytime you see anything for the first time, you mount an IgM response. Within 2 to 3 weeks, your body shifts that to IgG. So IgM and IgG are done. The only other thing I'll speak about in terms of immunoglobulins is IgA. Now, what about IgA? IgA is called secretory antibody, and it's present in, it can be secreted by cells that are lining your mucous membranes. What do we mean by mucous membranes? Mucous membranes are anything that's moist, and like, for example, the inside of your nose, the inside of your mouth, the inside of your foot tube, the linings of your stomach, intestine, all these are moist, right? And they are able to secrete IgA antibodies. Now, why is this important? So IgA antibodies can be secreted only from these mucous linings. IgG antibody primarily present in serum or blood. Okay, so they are different. I'll tell you why that is in a bit. I'll just tell you we're getting delayed a little bit. I'm just going to erase this. So you need to remember 2 classes. When you get a vaccine, after 2 or 3 weeks, you'll have IgG antibodies in your blood against that particular pathogen. Okay, IgA, I'll come to it in a bit. That's why I introduced these 2 terms. Now, you'll have antibodies. If you take the injectable poliovirus vaccine, you'll have IgG antibodies in your blood for very long periods of time, several years. Now, will this IgG in the blood prevent poliovirus infection? If you look at the poliovirus pathogenesis, where do I have it? Give me a second. Okay, here. So it enters like this. It replicates a little bit in your throat, a little bit in your stomach, shed in your feces, spills over into the blood, then it goes to the brain. That's how poliovirus works. Now, because you have antibodies only in your blood, when the virus spills over into the blood, that is when these antibodies will neutralize the virus, and they will not allow it to cross the blood-brain barrier. Once the antibodies bind to the virus in your blood, the virus becomes inactivated because these are neutralizing antibodies. They block the ability of the virus to bind to a host cell and get into a host cell. These are neutralizing antibodies, and you have them in your blood. So when you take the injectable poliovirus vaccine, it will not stop the replication in your throat. It will not stop the replication in the Peyer's patches in the small intestine. However, after that, when it spills over into the blood, it will neutralize the virus completely in the blood. That way, the virus cannot cross over to the blood-brain barrier, and even if it does, it's already neutralized. It cannot infect neurons. So that is how it prevents the major illness. It may not prevent a minor illness. It may not prevent a little bit of scratchy throat or a few things like that. It will not prevent those, but it prevents completely. It prevents bulbar or spinal form. That it will prevent very efficiently because it neutralizes the virus in the blood. Is everybody with me on this?
Yes, sir.
Yes, sir.
Okay, now let's look at the other second most common type of vaccine that we all know, we have all taken. That's called oral poliovirus vaccine, and it's a live attenuated vaccine. What is the difference between the first one and the second one? The two broad and the most widely used categories of vaccines fall into these two categories. One is the inactivated vaccine, where the organism is kind of killed or inactivated and given to your injected into your body. The second one is the organism is weakened or attenuated, and then it's given into your body. Now, how do you attenuate poliovirus? How do you weaken a poliovirus? So I'll just give you one line about this. We'll talk maybe when we talk about vaccines in detail, we can talk more about this. But how this was done, this was again done 7-8 decades ago by funding, with the help of funding that was initiated because of the poliovirus paralysis in the US. A lot of funding came, and that initiated this vaccination program also. How did they inactivate the vaccine? This vaccine causes disease when it's able to replicate in the neurons. All of us know that. This virus causes disease when it's able to replicate in the neurons. Now, this virus, when they forced it to replicate, for example, in kidney cells, it lost the ability, it acquired mutations. One change here, one change here, one change here. It acquired a few mutations that did not allow it to replicate well in the neuronal cells after that. They grew it in kidney cells for, let's say, 6 months, 9 months. They continuously grew it in kidney cells. A cell that it normally does not infect, they forced it to infect those cells. And so it acquired mutations to adapt to those cells. And then it lost its neurovirulence, or its ability to infect the anterior horn cells. It lost. It just expressed other sets of proteins that will be better suited for other cell types rather than neuronal cells. It just lost the ability to infect neuronal cells. And that is the beginning of a live attenuated vaccine. So they got lucky. I have to say that it's not easy always to do this. They tried it, they got lucky, and it worked. And it worked a big deal. It helped a great deal to eliminate poliovirus from the world. Oral poliovirus vaccine is much more important, has contributed in a much more significant way than the inactivated poliovirus vaccine. I'll tell you why in a minute. Now, when you take the oral poliovirus vaccine, I think in India, all of you have taken this oral poliovirus vaccine. How was it given? Anybody?
Drop.
Just drops.
Yes, sir, drop.
Just a couple of drops in your mouth. You're done. You're vaccinated. So it contains the weakened virus that cannot replicate in your neurons. What it does is that it produces what I refer to as local immunity or mucosal immunity. Remember, I told you mucosal immunity or the IgA. So that is possible with this vaccine because this is actual virus. It can be taken up by your tonsils when you get the drops. It can be it can replicate in the oropharynx and the tonsils. It can replicate in your Peyer's patches. It can spill over into the blood. but it cannot infect the neurons. So because it replicates in your throat and in your small intestine, it induces what we refer to as the mucosal antibody or the IgA response, which is typically primarily important for all the moist linings within your body. This is the best response that can help you. Now, because you have IgA response and you'll have memory cells and other cells locally in the region of your throat and in your intestine, when you're actually challenged with this virus, real virus, when you encounter the real virus, you're able to fight it from your throat and your gut. You will not even have any mild symptoms. Usually, not the throat symptoms, not the stomach related symptoms. You will not even have that because you have IgA response. Instead of controlling it in the blood with IgG antibodies with the inactivated poliovirus vaccine, here you're able to control it right here where it enters or in the small intestine. So you're able to control it at a much earlier stage because you have what is referred to as mucosal immunity or you have IgA response. Now,
Sir,
yeah, go ahead.
Sir, why it is not able to replicate in neurons when it have the same genetic material? You are saying that this is just the original virus. It is then why what then it should have the same genetic material as a virus which causes disease. Then why it is not? What's the reason it is not able to replicate in neurons?
It does not have the same genetic material. They forced it to grow in cells it normally does not grow. It acquired several mutations along the genome because they forced it to grow in kidney cells, for example, and they grew it for a long period of time. So it acquired slowly, it acquired one by one several mutations that allows it to grow well in kidney cells. And in the process, it accidentally lost the ability to grow neurons. So it has modified genetic material. It does not have the same genetic material, first thing. Now, how did they test it? They forced it to grow in other types of cells and they looked at the mutations. And then what they did is they tried to inject mice and see whether it causes disease. It did not cause disease. It did not cause paralysis in mice. And then they started slowly experimenting on a small set of humans and then a large set of humans. And that's how this vaccine evolved. Again, it is not possible to make vaccines like this on everyday basis. They just got lucky because this worked out perfectly well for them. For example, if you try to do this with SARS-CoV-2, you may not be successful. So it may be possible in only a handful of viruses. In many viruses, this type of work is not possible because you're talking about 1950s when this work on this vaccine was done. They didn't have much of any tools, but no mRNA vaccines, no DNA vaccines, no adenoviral vector vaccines. So they had only two types of vaccines. Either you can give a killed vaccine, which is the simplest and the commonest thing that anybody could do without technology. The second thing that they could do was to modify or weaken the virus. So these were the only two types of vaccines until mid 80s. We had no other type of vaccine. In mid 80s, we just added one more small type. I won't talk about it. So after that, until 2020, we did not have any other type of vaccine. We had only these three types of vaccines. Now we have two or three new classes of vaccines. But before that, only this was an option. So it has modified genetic material. Do you get that?
Yes, sir.
That is because of virus adaptation to the new cell type, you have these mutations. Okay. Now, what is the problem with this oral poliovirus vaccine? The problem with the oral poliovirus vaccine is that 1 in 3 million children who get this vaccine, the virus manages to reacquire mutations that will make it similar to the wild type virus, which means that will also have the ability for neurovirulence or the ability to infect anterior horn cells and cause paralysis. So this is a very rare phenomena, 1 in 3 million. However, 1 in 3 million children who receive this oral poliovirus vaccine are likely to suffer from some mild form of paralysis. So this is the flip side of the oral poliovirus vaccine. Now, what is the flip side? The ability of the virus to revert back, because it's a live virus. It can mutate and revert back. The other one here, it's a killed virus. It does not replicate in your body, so it does not revert. Here, this is a live virus. It can replicate in your body, so it can revert back to the wild type very rarely, but when it does so, it can cause what is referred to as vaccine-induced poliomyelitis or vaccine-induced paralysis. That's what it can cause because it was seen in a very low frequency. And this vaccine had several advantages over this vaccine. What are the advantages? What is the main advantages of OPV over IPV? Anybody?
It stops the
Does it translate?
replication in the intestine
Okay,
and throat
that is
itself. So
one
that's
thing.
what.
That is for each individual. That is a benefit. That is
You don't
correct.
need the specialist training to administer the doses.
That is a very important point. Now, to administer this, you need a trained nurse, and you can only do it at a certain pace because you need to clean the area. You need a needle. You need a syringe. You need, and you need to sterilize the area, and you need a trained personnel to inject it. Now, and you have to go to a hospital to do that. Whereas in OPV, all of you are aware of the Pulse Polio program, where untrained social workers, you don't need any training to just press a bottle and leave 2 drops in the mouth of an infant or a child. You don't need any training. So these are untrained workers who can just go around and just add 2 drops in the mouth of an individual. And this is done, for example, entire North India, it's done at the same time. Let's say January 13th, they'll just do it in entire North India. Now, we are not doing it that frequently anymore because it's been eradicated. They're not doing it much. But just even before 2-3 years ago, they would still do it one day, entire North India door-to-door campaign. You don't have to go to a hospital. All the children in your house, every house, there'll be social workers who come. If you have a child under 5 years of age, they give you the vaccine. So this they did door-to-door campaign was possible because of the oral poliovirus vaccine. Is simply not possible in a country of our size. If you have to inject it to, let's say, entire North India on the same day, it's simply not possible. You'll require millions of workers to do it. But whereas here it's much easier. You don't need anybody who's trained. So that makes it much faster and easier. And it also has another advantage that is kind of hidden. It allows for better herd immunity. I'll tell you what herd immunity is in just a
Sir?
bit. And then, yeah, go ahead.
Sir, what is killed virus? Sir, virus is anyway, it is not a living thing when it is outside the cell. So
That's why
then what's the meaning of killed virus?
Yeah, so we usually don't use the word killed, but I just try to use that word so that you guys understand it's not alive. But we use the word inactivated. By inactivated, for example, the nucleic acid material is all cross-linked so that it cannot replicate when it's inside the cell. It cannot replicate because the nucleic acid is rendered inactive by formalin. So it does not have the ability to replicate even and produce even a single virus. It cannot produce. So that is what we call as inactivated virus. I just use the term live and killed so that you understand what it is, but we don't use the word killed usually. It's inactivated.
So that's why it will remain only where it is being delivered physically.
That's correct. If you inject it into the deltoid of muscle of your hand, what will happen is there are some macro antigen presenting cells locally anywhere in your body, and those antigen presenting cells will take it up and it will not replicate anymore. But even the numbers that you inject are large enough that it's able to The antigen presenting cells will be able to attract T cells and B cells and mount an immune response just by the virus being in this area locally. Is that clear?
Yes, sir. Yes.
Okay, now I'll tell you what
Sir, actually, I have
Go
a
ahead.
doubt.
Yeah, go ahead.
So, so in case of inactivated virus, there is a requirement of a booster dose. Is it like that?
Okay, so that is yeah, good questions, many of these. So what happens is, simply to answer your question, if you use the inactivated virus, whatever dose you give to you, inject it into your hand, that number of viruses does not change ever, because not even one more virus is made. So that number is very important, and usually the dose is large. You need a larger quantity. But here in the oral poliovirus vaccine, because it's a attenuated virus, but it's live, it can infect your cells, it can replicate in your throat and Peyer's patches. Whatever number you given is amplified by the body because it can replicate in your body without causing disease. You need smaller doses or lower number of viruses when you use live attenuated vaccination, because it can replicate and amplify in your body. And the same phenomena is responsible for reducing the need for booster doses when you use a live attenuated vaccine. Does that make sense?
Yes, sir.
But when I say reduce the need, it doesn't mean that any live attenuated vaccine you need only one dose. There are some kinetics of immune response. When you see the same antigen for the second time, the amount of antibodies go up like 50-fold, 100-fold. So usually there are booster doses for both types of vaccine, but the dosage and the number of boosters are reduced largely for live attenuated vaccines. Okay? Is that clear?
Yes, sir.
Okay.
Yes, yes.
Now, there is no specific antiviral therapy available for poliovirus. It's only available for a handful of viruses and not poliovirus. I'll tell you what herd immunity is in a little bit, and then I'll explain the role of OPV in herd immunity when we do that. Now, what are the reasons behind the success of poliovirus vaccination? How have we as humans and this community, how have we been able to eliminate poliovirus from the most parts of the world? It's because of several reasons. We had two fantastic vaccines, very good efficacy, 95% plus efficacy, which means maximum only five out of 100 people will not respond well. 95 people out of 100 will respond well. OPV induces local immunity. That's again a factor. But there are some hidden factors that we don't really appreciate. And what are these factors? So one is that this is a fantastic vaccine. I already told you that. But these are some hidden factors. What is this one here? There are no animal reservoirs. I told you I'll come back and touch upon this in yesterday's lecture, that poliovirus can only infect humans. That's a very important aspect of why we have been able to eliminate poliovirus. What is the only other disease that has been fully eradicated from the world? Polio is almost eradicated, not yet, but one disease has been fully eradicated. Which one is that?
Smallpox.
Smallpox, right? And humans are the only hosts. Poliovirus almost there, humans are the only hosts. For example, the way I like you to think about this is, for example, if I had a fantastic vaccine that I made against dengue virus, will we be able to eliminate dengue virus from the world? We won't be, because there are animal reservoirs. Mosquitoes are reservoirs, and it's not possible to vaccinate the mosquitoes, at least as of today. So so that way you will not be able to eliminate diseases which have multiple hosts or animal reservoirs. For example, Ebola virus, fruit bats and other animals in the forest, they are reservoirs. So you cannot hunt down each animal and vaccinate it. So you will not be able to eliminate these diseases. But whereas if only humans can get infected, and if you eliminate, if you have 10 days where no human is infected in the whole world, it's gone. Even for coronavirus, if you have 0 cases in the whole world for like a week or 10 days, it's pretty much going to be gone, because after that, there will be no source of the virus that can spread from person to person. But coronavirus again has animal reservoirs, which complicates the story. So for any disease or a virus which has animal reservoirs is much more difficult to eradicate. Polio had no reservoirs, humans are the only host that made it possible. Now,
Sir,
this
in
part
case
right,
of a small,
yeah, go ahead.
sir, in
Yeah.
case of a smallpox, isn't it related to somewhat cowpox?
So it's not related to cowpox. Cowpox and smallpox are different viruses. Cowpox virus can infect cows, smallpox virus could infect humans. But we used the cowpox virus initially to protect against smallpox in humans because they shared antigens, they shared neutralizing epitopes. That was the birth of vaccination. The era of vaccination began with that, but they are different viruses. Okay.
Yes, sir. Yes.
Okay, they're completely different viruses. Cowpox does not cause disease in humans. That makes it simple, right? But it can help serve as a vaccine. Okay. Now I'm going to focus into, we have only 5 minutes. Okay. We are greatly delayed in today's class, but that's okay. I'm going to focus on this aspect in the next 5 minutes and finish it. What about this aspect of OPV and herd immunity? I'm going to expand on that. So maybe first I'll tell you what is herd immunity. Okay. Uh, Live attenuated vaccines such as OPV, they are able to replicate in the body, like I told you. Once it replicates, let's say I get pulse polio drops. Okay. It replicates and it's shed in the feces, and that can go and infect unvaccinated individuals. So if all the kids in North India under 5 years of age, if they get the pulse polio vaccine or the oral vaccine on the same date, today, after 3 months, after 6 months, after 9 months, if they keep getting this, what they'll do is, you all know that once you get infected with poliovirus, I told you yesterday, the shedding in feces happens for a long time. So these children under 5 years of age who got vaccinated will be shedding the vaccine strain in their feces for months together. And then they get the second dose of vaccine if you missed it. Then the third dose, then the fourth dose. They got multiple doses of this pulse polio. So constantly they'll be shedding off the vaccine strain in the feces, in the community, in the entire country. The whole country, all children under 5 years of age are shedding the vaccine strain in the feces. And that will also spread to other unvaccinated children. So for example, because of poor socioeconomic status and poor hygiene, fecal-oral route of transmission of the vaccine strain also happens. So unvaccinated children can also become protected unknowingly. They will not even know it. They will ingest the vaccine strain because everybody is shedding it. All children under 5 years of age are shedding it, and they don't know about hygiene, and they would be getting it. So all unvaccinated individuals, pretty much they become covered by mass vaccination at the same time. So what is the fundamental of herd immunity? Herd immunity is nothing but just the immunity in the community. For example, this is a person who's infected and transmitting. This is a susceptible person. He gets infected, then he can also transmit it to the next person. But if a transmitting person comes in contact with the immune person, that blocks the chain of transmission. And this is the most important thing. The number of such individuals you have in the community will reduce greatly the burden in the community. Or in other words, let's assume all these sheep are vaccinated, only this one is not vaccinated. Okay, this one is not immune, the black one. But because rest of them are all vaccinated, this black sheep is never going to be exposed to the virus because it's surrounded by people who are immune, who will stop the chain of transmission. So that is all about herd immunity. It's the immunity in the community. It's about protecting people who have not been able to get themselves vaccinated. Some of them may have conditions such as immunocompromised conditions that may not allow them to be vaccinated. But however, if this guy is immunocompromised, if everybody else around him is vaccinated, he's going to be good. He's going to be protected against poliovirus. So, like in 2014, I think this is Nigeria, if I'm right. This is Pakistan and Afghanistan. So only 3 countries. Today, this is off the map. We have only 2 countries in the world which have reported poliovirus. As of 2020, is only Afghanistan and Pakistan. The poliovirus has not been reported in any other country for the last 2 years except these 2 countries. I think we'll stop there and take questions. Next 2 minutes.
Sir, in case of viruses,
Go ahead.
sir, am I audible?
Yes,
Yeah.
you are.
In case of viruses, what does it mean by immune? Like, take for example, if I am immune, does it mean that if viruses are infecting me, I am able to kill the viruses? And if I am not able to kill the viruses, I am immune means I am even the viruses infect me and viruses come inside me, no effect will be shown any kinds of I will not get any kinds of stress, physical or any kind of. But as the viruses are, I have that viruses, can't I transmit to anybody else, even if I am immune?
Yeah. Okay. So again, it's all about a lot of our knowledge comes from coronavirus and vaccinated people being infected and being able to transmit. That's absolutely true for coronavirus. But what you have to go back and look at is the fundamentals of what I just told you just now, the difference between IPV and OPV. In at least in coronavirus, pretty much whatever you take, they're not necessarily inactivated virus, but they fall into this class that they will not elicit a mucosal immune response. Is that clear to you? None of the coronavirus vaccines that are approved today will induce IgA antibodies in your respiratory tract. None of them until today. Research is going on. They are not yet approved. So because you will not have IgA antibodies against coronavirus. You can still get infected, but because you will have IgG antibodies if you're vaccinated, it makes your disease milder by neutralizing the virus and preventing its spread and replication to a good extent. But because it can replicate in your throat and your respiratory tract for a little bit, you can also spread it. So there are different things. So primarily what you have to understand is only a vaccine like this. Let me clear this very quickly. Only a vaccine that is a live attenuated coronavirus vaccine, for example, it will be a nasal mist. You just have to inhale it for a minute and you'll be vaccinated. That will be the vaccine. That will be a live attenuated for coronavirus. For this, it's drops because it goes into your throat and GI. For coronavirus, it has to be inhalation, and they are working on it. So once that happens, then very few vaccinated people will be spreading the infection. Most of them will be able to control it in their throat. It cannot even replicate for 1 or 2 cycles because they will have IgA immune response. They will have more local immunity. They will control it replication in the respiratory tract before it can spread to others. There will be basically little or no replication if you have mucosal immunity and an IgA response with a nasally inhaled vaccine for coronavirus. Then you'll not be spreading it. So that is the difference. Does that make sense?
Yes, sir. If it cannot replicate, then there will be no transmission.
There will not be transmission, but we don't have a vaccine like that, and that's why you're saying what you're saying. But with oral poliovirus vaccine, that is exactly what happens. These children who got OPV, they will not be even transmitting. whereas the children who got IPV can be transmitting. You understand, right?
Yes, sir.
If they
So,
get actually, yeah, go ahead.
so not having an animal reservoir means, does it limit us in terms of experimentation as well? Because if the host is not infected, then our studies remain just limited to humans. So if we want to test the virus, do we do that on humans directly?
Okay, so that's a very good question. So what happens is when we say no animal reservoirs means naturally these animals are not infected, the animals in the wild. But you may be able to force a laboratory animal such as mice or monkeys or primates. You may be able to force infection, then kill the animal and finish it there. It doesn't go out of the laboratory. And even if the mice or primate does not usually have that receptor, you can also overexpress that receptor and force the mice to get infected and do your work in mice. Get it?
Sir, like,
Yes,
do
sir.
do we have some examples in which some virus is some humans are reservoirs for that virus, but chimpanzees are not? And if not, then why it is not? Like, I'm thinking that humans and chimpanzee had very less amount of, like, 1% of DNA difference. So
Okay,
like,
I'll
is
answer.
there?
Yeah, I got your question because we're running out of time. I'll answer it very quickly. I got your question fully well. So I'll tell you a very simple thing. Most hepatitis viruses that infect your liver, when you take any kind of liver cell, 10 different people, you take the liver cells, you try to force the virus to grow, it does not grow. It can infect you from your own liver cell in the plate, it will not grow. So it's much more complex than what you think. So you need a 3D architecture, you need a variety of things for the infection to happen that may not happen. It can infect your liver, but when you take your liver cells out, it will not infect you. So most hepatitis viruses cannot grow in any liver cell line naturally. It does not. So that's just a short answer to your question that why viruses that can infect humans may not infect another closely related animal. It may not even infect your own cells in cell cultures. It's much more complicated than you think. There is a lot about 3D architecture also that's involved. And your liver and my liver also may differ in those architectures, for example. So chimpanzee's liver would be very different for you. So that way, I hope that answers your question.
Yes, sir.
Okay, I think we
So
can
just
definitely.
one last question.
Yeah, sure. Okay,
So,
quickly.
like poliovirus was for the live attenuated form was made such that it could infect the kidney. So, when we actually give the virus, is there a chance of infection happening in the kidney or the organ that we used?
So they use monkey kidney cells. It acquired
Okay,
mutations, and
got it, sir.
it may not infect our kidneys most likely, and it'll replicate in the throat and the stomach. By the time you'll have some immune response, and to the best of my knowledge, it does not infect human kidneys.
Right, sir. I thought it was the human kidneys that we used. That's
No,
why
no,
I
it
was.
is they use monkey kidney cells.
Okay, sir.
Sure. Okay. Okay, guys. Thank you.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you. Bye bye.